Good afternoon, everyone. Thank you for joining us, and thanks to our generous benefactor sponsor, Mitsubishi Tanabe Pharma America. The program will begin in 2 minutes. Back soon. Good afternoon, and thank you for joining us today for the next installment in our Ask Me educational webinar series, sponsored by Mitsubishi Tanabe Pharma America. My name's Audra Hines, and I'm the Vice President of Care Services for the ALS Association Golden West Chapter. The chapter care managers continue to connect with our community via a variety of online mechanisms. We offer online support group meetings using Zoom, which allow you to experience all the benefits of attending a group from the comfort of your own home. We have 3 new topic-specific support groups that have launched or are launching soon, including Spanish language, familial ALS diagnosis, and a trach vent specific group. Remember to visit our chapter's online calendar for monthly updates, such as guest speakers, new meetings, and date changes. If you'd like to learn more about our programs and to support the chapter, please visit our website at alsagoldenwest.org. Today, we're proud to host this special program on how ALS research advances during the COVID-19 pandemic. Introducing our guest speaker for today is our President and CEO of the ALS Association Golden West Chapter, Fred Fisher. Fred?
Thank you, Audra, and good afternoon, everyone. Thank you for joining us today. By now, you're familiar with the common refrain, these are unprecedented times, these are historic times. But what puts them in context is not what difficult times do to us, but what we do in response. That's what living with ALS is about, and that's what finding treatments and cures is about. Seeking solutions every day, no matter the obstacle, to extremely challenging problems. Our goal is to overcome the extraordinary challenges associated with ALS care, services, challenges with public policy, and certainly research challenges. Regardless of the time, we remember what we did to overcome. And what we do today is built on the efforts of those who came before us. Today, we are building on the efforts of scientists, philanthropists, and public health professionals who came before us seeking to end neurodegenerative disease. The innovators, the dreamers, the doers, and today, collectively, we are those people. Our guest speaker today leads the way in terms of innovation, seizing opportunity, and an ability to bring treatments to the ALS community. He reflects the way we value the necessity of partnership with scientists and industry. We share a purpose and make progress towards our vision, creating a world without ALS. Steven Finkbeiner is the director of the Center for Systems and Therapeutics and a senior investigator at Gladstone Institutes in San Francisco. In addition, he's a professor of neurology and physiology at UCSF. He earned both an MD and a PhD in neuroscience from Yale University. He completed an internship in internal medicine and chief residency of neurology at UCSF, followed by a research fellowship at Harvard Medical School. Dr. Finkbeiner's expertise encompasses iPS cell modeling, human genetics, robotics, imaging, computational methods, and artificial intelligence. He's known for inventing robotic microscopy. He'll tell me how to pronounce it. a platform for performing high throughput longitudinal signal cell analysis, and for a series of discoveries about the biology of disease made with this approach. Steve is a member of the California ALS Research Network and the winner of the Golden West Chapter's 2017 Essey Commitment to a Cure Award. He's a principal investigator in the Neuro Collaborative, a research initiative launched by the Golden West Chapter to accelerate ALS therapy development, creating a virtual super lab in partnership with Gladstone, Cedars-Sinai Medical Center, and UCSD. Steve will also be hosting the chapter's 11th annual California ALS Research Summit in 2021. Please join me in welcoming Dr. Steve Finkbeiner.
Great, thank you very much, Fred. And let me also extend my welcome to everyone on this video conference. It's a real pleasure to be with you today. And as Fred said, we've had a long relationship with the ALS Association, and I think the Neuro Collaborative is an exciting initiative to help accelerate the development of therapies for ALS in coordination with drug companies. Today, though, I wanted to spend a little bit of time telling you about an exciting national project that around ALS that has unprecedented in its scope and scale. So just to make sure that we're all on the same page, I know for many of you this will be remedial, but I wanted to make sure that we didn't go too fast for anyone who might be new to this subject. So let me first start by just saying a word about what ALS is. So it's a progressive degenerative disease that affects motor neurons. And what you're looking at here in this figure on the right is sort of cartoons of different parts of the nervous system. And the important thing with ALS is that it affects the neural circuits that control our voluntary motor movements. When we want to move a muscle, the signal begins in our head in a part of the brain called the motor cortex. And there's a neuron there that will send a signal down into the spinal cord. And there is a second motor neuron that sits either in the brainstem, which is kind of around in your neck area, or farther down in the spinal cord. And that neuron in turn will travel outside the nervous system and connect with the muscle that it wants to control. And so when we want to move a muscle, a signal gets sent from our brain to our spinal cord, and then from our spinal cord to our muscle. And the problem in ALS is that these neurons, both the ones in our brain and the ones in the spinal cord, begin to undergo degeneration. And as that circuit gets broken, it becomes progressively more and more difficult to control our motor movements. And unfortunately, it's a fairly aggressive disorder, although the experience that different patients will have can vary quite a bit for reasons we don't fully understand. And eventually, some of the muscles that control our breathing are affected, and that's ultimately what causes people to die in ALS. So in terms of what causes the sort of underlying molecular causes of ALS, scientists generally divide diseases into genetic causes and non-genetic causes. In the case of ALS, about 10 to 13% of patients have a gene that we can identify with a mutation that we think is responsible for ALS. But most people who have ALS don't have a genetic mutation we can identify. In fact, although there still are patients who have familial forms of ALS and the gene remains unknown, at this point in ALS research, we actually know the majority of the cases with familial ALS, the gene that's involved. This pie chart here is meant to illustrate the major genes that cause familial ALS. The first gene that was discovered was a gene called superoxide dismutase 1. More recently, a gene called C9orf72, and I apologize, neurologists and neuroscientists don't pick names for genes that are very interesting sometimes, very hard to remember. But this mutation in this gene, it turns out, is the single largest cause of familial ALS. It also causes a fairly significant fraction of another neurodegenerative disease called frontotemporal dementia. And sometimes patients can have symptoms of both ALS and frontotemporal dementia. Smaller contributions are made by two other genes, FUS and TDP43, and then there are a whole slew of other genes that are rare causes of familial ALS. I do think TDP43 is worth just mentioning in particular, because although it's very rare for people to have a mutation in that gene, when we look at patients' brains with ALS, almost 98% of patients have abnormal collections of this protein, even though the gene itself may not be mutated. And so we think that this particular gene and protein may have a special role in ALS that we're still working to understand. So 10 to 13% of patients have a gene with a mutation that causes ALS, but that leaves a lot of people with ALS for whom no genetic mutation can explain the ALS. Now, that's not to say that there still isn't important genetic contributions to ALS. In fact, when we look at twins and we try to measure how heritable ALS is, it turns out that somewhere between 40 to 60% of the risk of ALS is heritable. So we think that there still is a strong genetic component, even if the ALS isn't explained by one mutation in one gene for those particular patients. In fact, kind of the way we think about it is people get dealt like a card game, a deck of cards, and if they have certain combinations of variants in their genome, that may make them at an elevated risk for ALS. And so that is a big focus for a lot of ALS research now, is to try to understand better those genetic underpinnings. And the better we understand those, the better able we will be able to understand what other contributions to ALS might be present. For example, many of you are familiar with certain proposed environmental triggers for ALS. Many of those haven't been very well established, but as we understand the genetic causes of ALS better, it'll be easier for us to be able to investigate those environmental causes with greater certainty. So that's the background. I wanted to explain the motivation for the research project that I'm going to tell you about by trying to highlight two major questions in the field that we're trying to understand and answer. So the first question may seem odd at first: is ALS one disease or many diseases? But you have to remember that for many neurological diseases, the way those diseases got defined was by a clinician who saw patients come in with a certain set of symptoms. And I think what we've learned as we've begun to understand other diseases better and better is that just because patients share the same symptoms doesn't always mean that they have the same underlying cause. And it turns out that's really important. The reason it's important is in part because of how we design clinical trials. The way clinical trials are done now, we assume every patient who has the same clinical diagnosis like ALS also has the same underlying cause for their ALS. But if that's not true, if it turns out that there are actually subgroups of patients with ALS, maybe a subset with a certain cause and then another subset with a different cause, that'll be a real challenge for clinical trials, because the way clinical trials are currently done, we enroll enough patients to be able to detect effects of therapies if everyone, in principle, could respond to that therapeutic. But if it turns out that ALS is a combination of underlying causes, and that in any given clinical trial, maybe only a subset of patients really stand to benefit from the therapy because they have the mechanism that that drug could work on, then that's a really big challenge, because what it means is that the clinical trial won't be successful because we simply won't have enough patients to be able to detect an effect in that small subgroup. So even if the drug actually works, it'll be difficult to prove using statistical methods that the drug has had an effect. So it's very important for us to try to answer this question. If we were able eventually to define or divide the disease up into subgroups, then that would make it possible for us to design clinical trials in a different way, where we might be able to just study one subgroup, maybe the subgroup that we think might most benefit from the therapy that we're trying to test. And just as a frame of reference, this is the approach that has been taken recently in cancer, and it's been very successful. Cancers, again, were oftentimes defined by the sort of origin of the cell type, like breast cancer or colon cancer. But as we've begun to subdivide these cancers, it's led to new treatments and much more effective clinical trials. And so this is a really important objective in ALS. The other has to do with really thinking kind of creatively and inventively about trying to develop new and faster strategies to treatment development. And one of the really exciting, I think, developments in the last decade has been how new approaches using very large data sets has made new strategies for developing drugs more feasible. Conventionally, what people do is they first try to understand the mechanism of a disease and then come up with a rational strategy based on that mechanism to develop a drug to treat it. That strategy has served medicine very well over the years, but it's challenging because it can take a long time to really understand mechanism well enough to target it. Sometimes people estimate it's 13 years to develop a new drug, and it can be extremely expensive. Some people estimate $1 billion or more per new drug. What we've discovered in the last, I'd say, 5 to 10 years is that there may be other approaches to this problem. There's a whole series of technologies that we collectively called omics, genomics, transcriptomics. Basically, these are ways to be able to look at sort of the universe of genes and proteins in a cell all at once, and then to use computational approaches to be able to see if we can get a kind of a big picture of what's going on in that cell. And what people have discovered is that with those sorts of big picture approaches, it's possible to essentially create a fingerprint for the effect of a drug on a cell, and likewise use that same sort of set of technologies to try to understand the effect of a disease, essentially the fingerprint of a disease on a cell. So even not without necessarily understanding the details, we may be able to get a really big picture of what's going on both in disease and in response to drugs. And there's been an effort at NIH now for a number of years to develop these fingerprints. So far, it's focused mostly on cancer, but to be able to create fingerprints of all the FDA-approved drugs, at least in cancer cells. And so the big idea is to try to bring this to a treatment strategy. And so the research project I'm going to tell you about today is called Answer ALS, and it's really looking for fingerprints of both ALS with these omic strategies. And in the same way that you can see a reflection in a mirror, or in this case, a city on a water, the idea would be if you could find a fingerprint for ALS and then find a drug that produces essentially the opposite fingerprint in cells, you might be able to use that drug to reverse the effects of the disease. And although this hasn't been done yet for neurodegenerative diseases, it has been done in a number of other disorders, certain cancers and skin diseases. And it's, I think, an exciting potential direction. And one of the appeals is that because these fingerprints are being created for drugs that are already FDA approved, you wouldn't have to go through the lengthy approval process that takes 13 years, and you wouldn't have to spend the $1 billion. In other words, if you found a drug that reversed the fingerprint of ALS, you might be able to test it in people very quickly because it already is FDA approved and passes safety tests. So that's really the big picture. We want to understand, is ALS one disease or many? And then we want to be able to use some of these advanced technologies to create fingerprints of ALS and then use computational approaches to see if we can find drugs to reverse that. So this is a really big collaborative research project that involves multiple investigators all over the nation working together. I think that's very exciting. And so what we've done is we've set up a whole clinical network to collect, and we've already finished enrollment of over 1,000 ALS patients in a variety of clinics in the United States. Those patients have been seen by clinicians, and clinical data have been collected both when they first appeared and over the course of their ALS. And at the same time, we've collected samples from them to be able to sequence their entire DNA. We've actually made iPS cells, induced pluripotent stem cells, from every patient who got enrolled. And we've been using those iPS cells to make motor neurons from those patients and to study those with some of the omics technologies that I mentioned earlier. And then, as you might imagine, this is generating mountains of data. And so we've had to use advanced computer techniques to be able to integrate those data to see if we can begin to understand our fingerprint. So we're well underway, but we're still far from finishing. We've finished our enrollment. My laboratory is responsible for all the whole genome analysis, which we have mostly completed. And then the iPSC production is underway, nearly done as well. And we're now at the phase where we're starting to generate these deep omics signatures. And unfortunately, did get delayed a little bit by COVID-19, but we are trying to do our best. So, as I mentioned, there are multiple clinical sites across the United States who are helping with this. You get a sense of that from the map here, and a sense of how many patients each site enrolled. And this has been just a terrific collaboration with clinicians who are very engaged with the basic scientists. I think we're all on the same page in terms of what the objectives of the program is, and also trying to make sure that the data we collect gets used maximally throughout the entire program. The DNA, once we collect it, is sent to the New York Genome Center. This is a center in Manhattan that was a collaborative effort between a number of institutions in New York, and they are part of a number of sequencing efforts for diseases, including other sequencing efforts for ALS. So one of the major advantages of doing this at New York Genome Center is that in addition to getting the sequence data that we want, we're also given access to data that others have generated that can be used to help us improve the analysis that we do. We already have begun to do analysis on the clinical progression of patients with ALS. In this upper left graph here, what you're seeing is lines of ALS FRS scores for each patient. For those who aren't familiar, the ALS FRS score is a rating scale that clinicians use to try to measure the severity of a patient's ALS. And you get some sense of how diverse and heterogeneous the clinical courses are. A lot of these lines cross one another. Some patients have more indolent progression courses, others are more aggressive. And one of the goals that we've had is to try to look at some of this progression data to see if it's possible to divide ALS patients up in terms of their progression rates. Perhaps that would suggest some underlying difference in the ALS that they're affected by. And that could give us the basis to start looking at our data to see if we can find some way to answer that question, whether ALS is one disease or many diseases. And so far, it has been possible to resolve courses of ALS into a number of subgroups based on clinical progression. And we need to do more work to be able to find out whether there's a real molecular basis for that. But even with data like this can be helpful for clinical trial design, because as we design clinical trials, if we compare patients who have more similar courses, it'll be easier and more sensitive in terms of our ability to detect the effects of treatments. We have, as part of this effort, we have a genetic counselor who is involved with patients. So one of the things that we really wanted to do was, as we collect the sequence data, if we did discover things that were unrelated to ALS that might help patients with their health, we wanted to be able to give those pieces of information back to them. And so we have Jennifer Rogenbach, a genetic counselor at Ohio State, has really helped our whole effort in figuring out ethical ways to be able to do this as quickly as possible, to both share with the patient who's participated in the trial data from them, and with their clinicians too, to help them understand and interpret the data. This has been a steady progress in both the collection and the progression of the program. This whole effort was substantially founded and funded by philanthropy, but in collaboration with the National Institutes of Health and the ALS Association. And it's largely being directed by a board from the business world, including a former senior executive at Caterpillar, a number of the airlines. And so this project is being driven in terms of milestones to be able to accomplish our output in a timely fashion. And in general, we've been hitting our milestones very well. In addition to enrolling samples, we've been generating induced pluripotent stem cells to be able to make motor neurons to study. And this project is really being overseen at Cedars-Sinai by Clive Svensson, who also is a member of the California ALS Network that Fred mentioned in the introduction. And this has been going very well, even as Cedars-Sinai is transitioning to a larger building to be able to carry out all this work. And I want to mention, I didn't maybe mention it earlier, but one of the other major objectives here was to not only generate data that we can analyze within our consortium, but to be able to make all these things publicly available. The iPSC lines that are made, all the data that we generate, as well as the computational tools. We want to make this a tremendous resource for the community to be able to really accelerate the discovery of causes and treatments for ALS. There are different ways to make a motor neuron from an iPS cell. We spent quite a bit of time within our group trying to figure out what would be the best balance in terms of the recipe we use to make a motor neuron between making the perfect motor neuron versus making something that was affordable at this scale to be able to do these experiments. And the name of the protocol that we came up with is called the Diamond Protocol. And that's been, I think, very well developed and refined. And again, steady progress, I think. So far, we've been able to continue the production despite the COVID-19. We've had some delay, but in general, it's been progressing pretty well. And one of our goals is to be able to create a repository for all the cell lines that are produced by Answer ALS. Right now, we already have, using the whole genome analysis that my group's done, been able to identify a variety of genetic mutations in some of these ALS lines, as well as other genetic variants that we speculate may change the risk of ALS for individuals. And we're busy using advanced computational techniques like machine learning and artificial intelligence to see if we can look at these genetic data to be able to understand better variants in other genes that may contribute to our risk of ALS, even if those genes when mutated, aren't known to cause ALS by themselves. And again, one of the goals here is to create a repository that will help accelerate and facilitate research in the broader ALS community. So these lines will be stored at Cedars-Sinai, but they'll be available to scientific investigators who can order these lines through this webpage using whatever search criteria that, that they want. So where we're at right now is these lines, the lines have been pretty much all reprogrammed. So we have taken the, for example, blood samples from patients and turned them into stem cells. We validated that those stem cells are high quality. And we're now at a stage where we're producing the motor neurons themselves to do this very deep characterization. One group is doing transcriptomics. They're looking at a molecule called messenger RNA in these cells. Another group is doing something called proteomics, which is looking at the proteins in each of these cells. Another is looking at something called epigenomics. And this is really has to do with how genes are controlled in the nucleus in terms of their regulation. And then my group, in addition to the whole genome analysis, is doing a special form of imaging that Fred pronounced so well at the beginning of the introduction, robotic microscopy. And the important thing here is we use robots that are capable of working around the clock 7 days a week. And this special form of imaging lets us identify and track individual motor neurons from patients, much like we would a clinical trial. And that makes it much more sensitive than we normally would. As I mentioned, my group also is doing the whole genome analysis. And we take that sequence data from the New York Genome Center. We have a computer pipeline that will go through, look carefully at the billions of nucleotide sequences of each patient, be able to find those that are different than the sort of genetic sequence that most people have, because those might give us clues about what might be contributing to ALS. And then we are able to look at those data sets all together. And then lastly, using very fancy computer tools, be able to integrate all these different data types with the goal of trying to see if we can generate that fingerprint for ALS that I mentioned earlier. And hopefully we'll be able to have more to tell you soon as this work gets farther along. At this point, we have already just done a smaller analysis to be able to test out some of the approaches that we're doing that's focused on C9orf72, that gene that I mentioned earlier that is the most common cause of ALS and frontotemporal dementia when it's mutated. And we were delighted to see that as we did all those omics types of experiments and looked for changes in these cells, a number of pathways showed up that have been linked in one way or another to ALS. I think the strength here is that this has really given us a kind of integrated picture of the biology. And one really interesting insight, I think, is that we've identified a number of pathways that we think might be responsible for contributing to or causing ALS. But at the same time, we've also identified compensatory pathways too. And I think that's a really important reminder that even though we tend, when we're thinking about disease, to focus on the thing that's really causing the disease, it's useful to keep in mind that our bodies often are trying to really fight the disease. We have a lot of pathways that are there that are really trying to resist neurodegeneration and make this situation as good as possible. And I think what's exciting to me about that picture is it really opens up two possibilities in terms of a treatment strategy. We can try to focus on things to block the causal factors, but at the same time, we might also be able to think about clever ways to boost the compensatory pathways that already exist in our bodies. We've taken these large data sets and have been able to compare differences in pathways and processes between ALS patients and controls, focusing first on C9. And we see a lot of biological processes really emerge from this analysis. And that's encouraging to us because it suggests that we really are onto something here. In addition, we're using other models besides human stem cells to be able to test the effects of those genes to get even more confidence that they really are real in terms of the biology. So that gives you a taste of sort of the science behind this project and the goals. As I mentioned, we're completely committed to making this data publicly available, as well as the tools and the computer resources. And so we've created a web portal to be able to help scientists in the field access these data. And we're trying to make it as easy as possible for them to find the data they want and to be able to receive it. And so that's really been the goal with the design of this web portal. We also, in addition to the work we do within our consortium, have reached out to a variety of other both scientific academic investigators as well as companies to help partner with us and be able to make this as useful and as powerful as possible. And we've been delighted to have Microsoft as a partner now for quite a while. Initially, they gave us quite a bit of space to be able to store data. And as you might imagine, a project like this does require a lot of data storage space. But in addition, they've really committed a number of their engineers to help us develop web portals and software tools to be able to analyze these data. And they're just one of many companies that we've interacted with. And I'm really excited. I think this is a great month to give this talk because it was this month we had our first public data release, and by far the largest data release ever of ALS biological information, and it'll be the first of many. And because the thing we care about most is to try to accelerate the discovery of causes and treatments of ALS. These data are being made public even before the academic investigators themselves get to really fully analyze or publish it, because we don't know who's going to come up with the solution of ALS. We sure want to do our part, but we want to engage the entire world to find the smartest people to help us with this problem. We've been busy. We're in the process now of really working on some of the computational parts of it, and I think we have an exciting kind of next year to roll a lot of this out. I think one of the great things about some of this computational work is that it can happen even when we have these shelter-in-place orders. So even though it's been difficult with social distancing to be able to do wet bench work, this is something that a lot of us can do remotely and at our computers. Lastly, I just want to say that I think the theme of this research project is collaboration. The number of clinicians and basic scientists who are working together on this is really extraordinary. And I think there is a genuine spirit behind this of generosity that really, with a clear goal in mind. And so I think that along those lines, we're reaching out as well to other government agencies, nonprofits, to be able to do this even better. As I said, the ALS Association was fundamental in the early stages of this project when we were doing work with NIH to be able to actually add the whole genome analysis component to this project, which was very important. So I think our attitude has been that we want to stay focused on the goal, and whoever shares our vision and goal and values, we're really eager to try to work with them. So I'll end there. I just want to highlight that, again, this has been a massively collaborative project. Clinicians and basic scientists from many institutions across the United States, as well as some tremendous supporters from the philanthropic space. So again, thank you for this opportunity. And I guess I'll turn it back to Audra in case there are any questions that people have that I might be able to answer.
Wonderful. That was amazing, Dr. Finkbeiner. Thank you so much. I am confident I did not understand all of that, but it was fascinating. And we have lots of questions to go with it. We had some questions submitted ahead of time, and I know some of you have submitted some live questions via the Q&A feature too. So we'll get to as many as we can. And if there are a couple that we can't get to, we'll certainly follow up with you with an answer. So the first question is, can you please comment on the recent Harvard study on gut bacteria and the role they play in the onset of ALS symptoms?
Yeah, so the question So the question pertains to the gut sort of brain axis. And for those who don't follow this area, there's been a lot of interest in the neurodegenerative disease area in the possibility that the bacteria that reside in our gut may play a role affecting the risk of developing certain neurodegenerative diseases. As I mentioned earlier, a fraction of patients have mutations in genes that by themselves seem to be sufficient to cause ALS, but that's still a small fraction of ALS patients, maybe 10 to 13%. There's still a larger heritable risk, but there's after that still maybe up to 50% of ALS we really can't explain. And I think people have generally been looking toward factors in the environment that may influence our risk for ALS. And so one of the ideas about an environmental influence is actually the bacteria in our gut. These bacteria, depending on which ones we have, may be able to stimulate inflammation in the gut that may be important for triggering certain neurodegenerative diseases. And ALS is not the only one. There have been other disorders, especially Parkinson's disease, where this is also featured prominently. And I think one of the attractions of trying to really understand that better is that there are ways that already exist to be able to manipulate the composition of the bacteria in our gut. So if we understood better what role they play, and if they really do contribute to ALS, then there may be opportunities there for developing therapeutic strategies that could be developed pretty quickly.
Okay, wonderful. Thank you. How you touched on this a bit in your presentation, but we got this question a few times, so just circling back to it. How is COVID-19 impacting current research, treatment, and clinical trials? And how is it stalling the studies on better treatments and cures?
Yeah, it's been a little uneven across the United States, but I can speak to our experience here in California and here because of the orders that we've had to shelter in place in the Bay Area, and then more broadly, state of California restrictions. What we've done at our institution has been to try to really limit the amount of work that happens at the lab itself. We've identified a handful of people who are doing essential work, and we've tried to be very careful in the experiments that we do and prioritize the ones that need to be completed. For example, ones that may have begun before COVID-19 that we want to complete or risk losing the data. But otherwise to try to respect the sort of overarching advice that we've got and keep the operation as focused as possible. At the same time, groups like ours, as you might imagine, do a lot of work on computers, a lot of analysis, and we do have lots of data. And so we do meet regularly through Zoom calls like this one with other scientists to be able to continue that data analysis and describe it. So some things have slowed down, other things continue apace. And I'm glad, our institution right now has begun to develop re-entry plans. And so hopefully soon we'll be starting to ramp up.
Okay, that makes sense. Thank you. Next question is, do you foresee neurone therapy becoming available to people diagnosed with ALS in the near future?
Is the question stem cell transplants or?
I'm not sure.
I guess as they're asking about the neurone trial, Brainstorm's trial of neurone.
I see. Yeah, I'm not directly involved with that, so I probably shouldn't say at risk of giving you bad information.
Sure, that's fair, no problem. This comes from a healthcare provider. Any indication when we can begin enrolling patients into clinical trials? Oh.
Yeah, boy, that's a great question. I think, one of the big issues with all of this has been, we've never really been through this before. And so a lot of this has been kind of make it up as you go along, where we're trying to use general advice in terms of epidemiology and public health to guide the decisions we make. but I think we're all very cognizant that time is of the essence when we're talking about ALS. And so I think each group is trying to think creatively about how can we move forward with the work we're doing, or the work we want to do, I should say, in the safest possible manner. And I think, I don't think that I have a good answer for folks yet. I think the other issue is, even with our reentry plans right now, there is a feel to it of take one step at a time. We want to begin this reentry process, see how it goes. And I think if people feel like it's working and we can proceed safely, I think you're going to start to see these things come back, as quickly as possible.
Makes sense. That makes sense. Is there any research currently with regard to a mutation in the VCP gene?
Well, yeah, so VCP, villosing-containing protein, is a gene that has been mutated in neurodegenerative disease. And so people have been very interested. They think that that gene plays roles in protein trafficking. It's not a common mutation, but potentially an informative one in terms of helping us understand mechanisms of disease. We think it may play. So one common thread that has emerged in ALS and other neurodegenerative diseases has been the idea that in a number of these disorders, proteins can misfold. So you can sort of think of it like origami. If you fold up a piece of paper a certain way, it makes sense. It's a rhinoceros or something like that, but you can easily misfold it into something that's useless or maybe even harmful. And so in the body, that can happen to your proteins too. So they have to fold up a certain way, and then they're very useful, but they can misfold as well and actually be harmful. And you may even have heard of certain names. People talk about amyloid plaques or neurofibrillary tangles or Lewy bodies. Those are misfolded versions of proteins in Alzheimer's and Parkinson's. ALS has its own name. It's called Bunina bodies, and they're basically these misfolded proteins that happen in ALS too. So we think VCP may be playing a role in part in dealing with that pathway.
Okay. And this is a question, I think, related back to your presentation. Does this research apply to related conditions such as PLS?
Yeah, we think so. So I think that one of the things that we observed as we enrolled patients was that people had a variety of symptoms when they were enrolled. Some had some frontotemporal dementia symptoms, some fell into sort of different categories in ALS. And so one of the things that we tried to do very carefully was to be able to record those details when people were assessed clinically. And the idea would be that as we begin to understand some of the omics data that we collect from cells from those patients, we can go back to the clinical data that we collected. So if we discovered that someone has a symptom profile that looks more like PLS, and they have a different kind of fingerprint for their disease than some of the others, that may give us really for the first time insights into a distinct cause that might help us define those patients and ultimately lead to better treatments in clinical trials.
Okay. And a related question from the same person, with the different courses you identified, how early can you tell what course an individual patient is on? And looking back at the data, you can see patterns, but how do you know early who might be part of a particular clinical trial?
Yeah, that's a great question. We need to collect enough data to feel confident about the trajectory someone's on before we, ideally allocate them to clinical trials. But we can't wait too long, or else we might have missed an important opportunity. So that's a terrific question. The way this study was structured with Answer ALS, we asked patients to come back periodically for assessments. And some came for all of the appointments, some came for some of the appointments. And the general sense was that with data from 2 visits, maybe separated by a month apart, we had enough to be able to be pretty confident about what trajectory they were going on.
Okay, wonderful. Can homeopathic remedies like acupuncture or acupressure prevent ALS expression for people with familial history?
As a scientist, the way we generally try to answer questions like that is doing special trials where everyone's blinded to whatever therapy they're receiving. And then we, including both the patient and the clinician, we do that because what we've discovered is the brain has this very powerful mechanism called the placebo effect, where people can literally, if they believe enough in a therapy, actually respond in some sense. And so we do that to make sure that the results we get really deal with the biology and not these potential confounding effects. I'm not aware of any data that show that those interventions would make a difference. And I'm not sure it's been tested, to be honest, in a rigorous way.
Gotcha. Okay. Another question, is international collaboration a significant element of this line of research?
Yeah, what a great question. It's kind of a sore spot for me because clearly ALS is a worldwide problem. And as are a number of these neurodegenerative diseases, and yet it's disappointing actually how little cooperation there is, at least at the governmental level. I mean, you might imagine governments would get together and kind of pool resources to be able to tackle these problems, but in general, they don't. I think for political reasons, mostly people want to support people in their own countries. Fortunately, scientists aren't themselves prevented from collaborating with people in other countries. And so what you'll often see are scientists reaching out to investigators in other countries to work together. But the funding opportunities are more limited. That said, foundations like the ALS Association and others don't have that same limitation. They can really fund the best research, and if they see ways to be able to bring investigators together, even across borders, they can do that, which is, I think, a real strength. And I think increasingly that is going to be a really key role that philanthropy can play, is to be able to harness some of the best minds all over the world and really, make the problem the main focus and find the best team to do it.
Okay, wonderful. Let's see, how about this one? Do ALS diagnosis rates match population growth rates? And is ALS prevention key to reducing complications after diagnosis? Yeah.
So the answer to the first question is a little complicated because there are a couple factors that govern incidence rates of diseases as they're recorded. One has to do with how sophisticated doctors are at diagnosing the disease. So for example, certain diseases, if they get more attention publicly, they become kind of more in the consciousness of doctors. And if patients first go to doctors who aren't specialists, then having that in their consciousness is really important for getting a diagnosis early. And that's one of the issues, I think, with ALS. We really want to get patients diagnosed as early as possible because we know time's of the essence. And so we want to be able to intervene as soon as we can. Another is that ALS, like several other neurodegenerative diseases, is a function of age. So although patients can get ALS at many different ages, they tend to get it later in life, 50s, 60s, later. And so we know that aging itself is a risk factor for neurodegenerative diseases. So the incidence of ALS is going to be partly a function of doctors' awareness and their diagnosis, partly a function of the size of the population, as the person who asked the question pointed out, but also partly a function of the age, the average age of the population. And one of the things that's been changing in the United States and a number of other developed countries, Western Europe, even China, is that the average age of the people in the population has been growing older and older. So we would predict, just based on that alone, the number of patients with ALS is going to go up. I've forgotten, Audra, what was the second question?
I will tell you. I'm gonna go back to it because we have a couple of other questions from that same person. So hopefully we'll get it all answered together. No problem. A question that goes back to your presentation. How would this program fit with a multi-drug study? And then they ask, UCSF has one in the plan for the next 6 months, I think.
At this point, this particular research project isn't connected directly to any clinical trial, but our hope is that if we can use the data from studying the patients in Answer ALS to find fingerprints of ALS, then what we might be able to do is, like I said, identify FDA-approved drugs that could be repurposed to perhaps treat, that might reverse the fingerprint of ALS for a subgroup of patients. If that happens, one of the wonderful things about having this great clinical group who helped us enroll patients is it's led by a group out of Mass General Hospital that's responsible for some of the major clinical trials in ALS as well. And I think there's a lot of enthusiasm for moving something pretty quickly from a result that we might get from a study like this into a clinical trial. And I think they're developing some very clever ways to do clinical trials in ALS. This is separate from Answer ALS, but I think it's worth mentioning. And it's a nice illustration of the idea that, everybody kind of working on different parts of this problem are really trying to improve each part, and hopefully it'll lead to an improvement overall. So one of the challenges with clinical trials is every time you want to do a clinical trial, it's a pretty long process. You have to, describe the trial you plan to do, you have to enroll the patients, you have to enroll a certain number of healthy patients and patients who are control patients with the disease who don't get the therapy, patients with the disease who do get the therapy, on and on. And so it takes a long time to set that infrastructure up, to enroll all those patients, to get it all done. And I think the thinking now is that for a lot of the therapies that we look at with with ALS, some of the measurements that we make are very similar. And so they've come up with a really clever idea of being able to more or less set up an infrastructure to be able to do clinical trials all the time. So you don't have to set it up each time for each clinical trial, but just have a infrastructure that's in place to do clinical trials, and then make it possible to be able to share control groups. So you don't have to have as many control patients too. So I'm really excited about that. I think they estimate that they may be able to speed clinical trials up tremendously, as well as cut the cost. And I think all those things will be very helpful for enabling us to test more things and to be able to do them in a really rigorous way that gives us more confidence when the results come out.
Okay, terrific. I'm going to wrap it with a final question. We got this from 2 or 3 different people who had a family member who passed away due to ALS, and they are interested in getting involved in some research themselves, just having that family history. What's a good place for them to find a trial in their area potentially to do that?
Well, so these would be people who don't have ALS themselves, but someone in the family passed away?
Correct.
Yeah, a big question really is kind of what would be appropriate for them. And I think that does take a little bit of discussion. There are, clinicaltrials.gov is a good place to go if people are just looking for an overview of the different clinical trials that are underway in ALS. There are fewer opportunities for people who don't have ALS and who aren't in particular risk for ALS. We do try to, for example, in this Answer ALS study, we do try to enroll oftentimes family members, unaffected family members as controls for that study. But there are relatively few opportunities for folks in that situation unless they have a family member who has ALS at that time and might be enrolled in a clinical trial.
Okay. Wonderful. Well, thank you so much. This was amazing information. You answered a ton of questions. Your presentation was so chock full of good stuff, and we couldn't have been more pleased to have you here today, Dr. Finkbeiner. I'm going to pass it back to Fred Fisher for a couple of minutes, and then I will wrap it up.
Thanks, Audra, and thanks, Steve. That was fantastic. And I think that your presentation combined with some of the questions that have come in really help illustrate how everything we do with the Golden West chapter supports the search for treatments and cures. And while that's not always clear to people, today's presentation really illustrates it. When Steve talks about biosamples, he's talking about people with ALS donating their blood, donating their skin. And it's those early engagements in research that are essential now more than ever. The advent of personalized medicine and the deployment of personalized medicine to accelerate therapeutic discovery, to accelerate the understanding of the disease, really made this possible. And it's what drove us to establish the Neuro Collaborative. And so to get people to donate their tissue and blood to science, you need an outreach effort. You need to be able to actually connect with the ALS community. And when you connect with them, you can't just invite them to donate blood. They have real issues of living with the disease every day. And so supporting people with ALS and offering those support services is an outreach opportunity to then engage those people in ways that they can participate in research. And of course, their story, your personal stories are what powers advocacy. It's your personal story that powers your fundraising, whether it's for the Walk to Defeat ALS, the Ride to Defeat ALS, whatever fundraising you're doing, it's going into supporting a system that's designed to find treatments and cures. And that advocacy, your stories power research funding, both private funding that we deploy directly to scientists, as well as the largest governmental source of funding is created by the ALS community coming together at our Advocacy Day, which is coming up at the end of this month. And you can find out more about how you can participate about that on our website. Because of your efforts, because of this integrated effort of providing people with service, engaging them in research, telling their story, the ALS Association is the largest nonprofit funder, certainly in the US, if not the world, supporting 175 researchers in more than 8 countries. And so it's your advocacy, your story, your support of what we're doing that is really powering an international effort. And we're so grateful to have so many amazing scientists like Steve on our side, working with us, helping us accelerate the search for treatments and cures. So it really takes all of us to be part of that. And we're very grateful to all of you for being here, and in particular to Steve and his team for not only the work they do, but for their work with us and willing to share their story with all of you. So thank you, Steve.
Thank you so much, Fred. I want to thank you all for joining us today. A special thanks to our generous sponsor, Mitsubishi Tanabe Pharma America. And as Fred mentioned, the Golden West chapter will be participating in the 2020 National ALS Virtual Advocacy Conference. It's virtual this year, so attendees will learn about the latest in ALS research, care services and advocacy, and leading all up to a virtual day of action. Together, we really can continue to fight ALS by educating policymakers, driving action to increase funding for research, accelerating the search for treatments, and improving access to care. And once again, as Fred mentioned, you can find out more about that on our website, also goldenwest.org. And if you too would like to support this free educational programming like you participated in today, please consider making a donation today at give.alsagoldenwest.org. And please watch for the short survey on today's presentation that you'll receive via email in order to help us plan for future programs like this one. Thanks so much again, and have a wonderful day.